Good morning, everyone. I'm welcome you all to this 11th day of CSIR online coaching. Before starting the section, I want to confirm am I audible? Whether my slides are visible? Please let me know. Am I audible? And can you see my slides? Okay. Thank you for your response. Now we are entering into the session. Okay, today I'm going to cover the methods in biology. In this topic, previously we discussed molecular biology techniques. In that topic, I missed three subtopics. Some of them asked to teach that particular small topics that are the each and every topic they want to understand. So only I am just explaining this in this slide. Today, as per the schedule, today's topic is immunological techniques, biophysics techniques, and radio labeling technique. Okay, before entering the immunology technique, just a few slides are there to explain what happened, what is the some molecular biology techniques. Okay, what is mutagenesis? Mutation happens. Mutation means that may be the change or deletion or suppression. Okay, in the in vitro mutagenesis, they have a two different type. One is the restriction fragment based detection. Another one type method of mutation is nucleotide removal. Okay, in the first type, that restriction fragment site, each and every gene they having the different different ends in the different restriction fragments. In the first explanation, they explained with they are explained with the band. This uh, full restriction fragment having two proteins. That two protein, the normal gene without mutation, that having the two ALU1 two restriction sites, that active the restriction site for ALU1 enzyme. Okay, in the experiment they are remove that particular restriction fragment. So after that after the experimental detection, there is only one ALU. It's a restriction fragment deletion. In the deletion, normal gene, they have a two restriction sites for ALU1. They are mutated, so there is only one ALU restriction site that is happen at the restriction fragment, the particular portion. So that after that mutation, there was a modified protein detected in the sample. Another one type is nucleotide removal type. The nucleotide particular portion is totally removed from the restriction site. So that is another one. In this step, they explained with the ECOR1. After the after the restriction, there was after they are divided into two portion, right? Every restriction enzymes cut the gene, so there was a two different portions. That two different portions are formed with another gene with ligase enzyme. That newly formed gene called as the small is a mutated one. There was some mutation happen at the restriction site of ECOR1. These are two types in the mutagenesis, that is restriction fragment deletion. Another one is nucleotide removal restriction. Another important thing is site directed mutagenesis, the advanced techniques of mutagenesis. In this mutagenesis, we may substitute the gene, we may delete the gene. Another thing is we can insert the gene. Insert means here substitute means remove and add. Remove the one gene at a particular site and add the new gene. Here insertion means with the native gene we need to add the extra characters. That is called as insertion. Here they having the three different steps. First, after the this mutation experiment, we want to amplify. With a small quantity, we are not able to do the experiment, and we are not that is not not usable for the future publication. So we want to get more get more amount of mutated fragment. We are doing amplification, PCR amplification. For that they are doing the hot star PCR. In the hot star PCR, after the hot star PCR, they are receiving the higher quantity of the gene. Then that treatment and enrichment kinase, ligase and DNA polymerase enzymes, because after that mixture is phosphorylated with the phosphorylation happens in the multiplied sequence, then that is ligated with the ligated each other. After the ligation, that template want to develop for full genomic DNA formation. These four steps are happen in the in the second step after the amplification of site-directed mutagenesis happened DNA. So on sequential experiment, just to understand the concept for laboratory level, if you want to insert for example, if the plant is we want to grow a plant in a fast manner, we want to take the gene from the fast growth having plant and insert into the slowly growing plant. That after that the further process we can do the mutagenesis and amplification and enrichment. Enrichment is the it's improve the ability and for its concluded form. Okay, next is the it's one type of technique widely used in the advanced genetic engineering labs, advanced molecular biology lab. They mention, so someone has specifically asked this portion, explain the what is gene knockout, what gene splicing. What is gene knockout means? It's typically removed the gene expression, fully removed the gene expression. There was no traces are there. Okay. It takes much time. Another thing, that's DNA silencing means we are not removing the gene, we are silent, we are suppress the expression. We are we can reduce the expression of the gene or masking the gene expression with the addition of anti-nucleic acid analog. Okay, it's expensive method for some of the advanced genetic in laboratories, molecular laboratories, they are doing the gene knockout in laboratory scale, and they are applied for the many medicinal purposes. So gene knockout is fully deletion of removal of the gene expression, but seeing the gene silencing means it's a by addition of the synthetic antisense nucleotide in the gene, we can silence, we can reduce the expression of particular gene. Okay, next is another one example. Here the gene knockout happened in the animal. In the one mice, they it shows a fully black color. So only one place having the white color. While injecting the recombinant that cells into the this mice, we can get the this chimeric mice. Chimeric mice having the both the recombinant character and the native character. That is the gene knockout. Only one expression completely stopped. So here some character is blocked, only the colors alone expressed. Okay, after that another one topic is the post translation modification. Okay, post translation modification, why it is important means every genome having the transcription process that after the transcription process that receives the mRNA, that received mRNA is undergone the translation process to produce the protein. Formed proteins are undergone various chemical reaction, phosphorylation, acetylation, methylation, various chemical reactions, natural chemical experiments happen in a human body. Due to this chemical process that are modified in itself, one protein can modified into uh, thousands of proteins. That is the post translation modification. After the translation, it's undergone the methylation, acetylation, that kind of chemical reactions. This chemical reactions change the structure, produce the structure and change the function and uh, structural and functional change on the protein explained with the post translation modification. How we can understand, how we can analysis in this post translation modification understanding, we are having the different steps that are explained with the biological sequential experiments. Okay, this workflow is very important. Why I am after the long search, I got this image because I'm not expect this image. I'm searched for the what is the protein post translation protein modification. That is kind of the search I did in the my laptop. After there's some period I am recognized this flow chart. It's a one very important flow chart which helps you to understand what are the methods in molecular biology. First is a sample collection, then is a sample preparation. Here in this sample preparation, you can do the cell disruption, protein solubilization, protein depletion, enrichment, removal of substance. These are all the basic experiments which helps to improve your research quality, improve your results. Then protein. Now you have the protein mixture. Someone asked the question the 2 days back, how to separate the protein. In the RNA and DNA and RNA are the genetic materials that each and every RNA isolation we have different method, protein isolation we have different method. Likewise we can obtain the protein mixture, then go for the various experiment. Okay, first experiment, that is a bottom up approach. First we want to do the STS page. After the STS page, we want to do the proteolysis process. After this proteolysis process helps to obtain the protein sample, crude protein, that purified protein sample. Then we want to do the reverse phase HPLC column. This reverse phase HPLC experiment is a very important thing for understanding the protein character. Okay, whether it is how what is the molecular weight, that kind of things we can understand. Then MALDI-TOF, it helps to understand the structure of protein. MALDI-TOF helps to understand structure of the protein. These are all the techniques helps to do your post translational modification. Okay, how to understand the post translational modification with HPLC? First, every proteins having the unique character. That unique character explained with the peak value. That okay, this protein having the peak at this point. That is the explained one. But if there is a change in structure, we are get the peak at various nanometers. That one way. Another way is molecular weight change. That in the STS page itself we can understand whether it is modified or not. Okay, these are the things which helps to do the protein analysis. Protein analysis for understanding the peptide uh, understanding the protein protein molecular weight and character molecular size weight means we want to do the reverse phase HPLC, that is RT HPLC. Another thing is the structure understanding, we want to do ESA or a MALDI-TOF. Okay, in this after the in each and every step we want to do the these are only some important tricks, that is the lipid estimation, carbohydrate estimation. That is the one thing for every cells having the lipid and carbohydrate, right? In the once you destroy the cell, the internal portion having the that extracellular membrane having the polysaccharide, and that polysaccharide can be hydrolyzed by buffers and enzymes. After the hydrolysis you can get the carbohydrates. Another thing is the cell membrane also having the lipids. How to separate it? If you are destroying the cells with the water molecule, means that polysaccharides are soluble in the water. So unsoluble, that insoluble molecules are called as the lipids and cell debris. After the next step we can obtain the lipids. Okay, first step is the biological materials collection, then doing the extraction. Here they did the extraction with the organic material, that solvents, or that these are all the highly the methanol is a mid-polar compound. The polarity index is 4.5 something, 5.1. Then these are all help to separate the lipids. Then for confirmation we can do the LCMS. After the LCMS analysis, these are all the techniques for the separation. Once inject into the lipids into the reverse phase liquid chromatography, we can obtain the separated, so we can separate the various kind of lipids. Then ionization process and deduction by the various MALDI-TOF experiments. This is another workflow of the separation of lipid. Here this is one of the experiment for estimating the carbohydrate, widely used experiment. Another techniques is here they are using the glucose oxidase peroxidase reagent, but some of the experiment they are using the DNS assay, so phenol sulfuric acid assay. This why I am mentioned this means they are having the question like that. Okay, if you are they are given paragraph for the carbohydrate extraction. Finally they are asked the question with how do you understand how do you identify the quantifying the carbohydrate. That kind of question they asked in the CSIR examination. So I mentioned this. You can calculate, we can quantify the carbohydrate with the DNS assay, phenol sulfuric acid assay, that are directly and indirectly help us to quantifying the carbohydrate. Here another method is the GOPOD reagent. First we want to take the starch, that is the polysaccharide. Then starch solution is digest with alpha amylase. That alpha amylase convert the disaccharides. That disaccharides are converted into the monosaccharides by the glucoamylase. These monosaccharides are quantified by the GOPOD reagent. Okay, next we are entering the our today topic is that immunology techniques. In this immunology technique, we want to understand the antigen antibody. First, we previously we discussed in immunology part. One time I'm going to just a refreshment. Antigen, which is the foreign particle. It may be the signal for the human cell surface receptor or antigens. The antibodies are present in our human cell, the self defense system, that are recognizing, once they are recognizing the foreign particle, it's activated. It's a basic concept. In this antibody generation experiment, they are producing the antibody in laboratory scale itself. So how can we produce the antibody? Okay, this each of B cells are only produce the antibodies to one epitope. Epitope means it's an antigen. Okay. Here the first image itself they mention the large immunogen field multiple epitope polyclonal antibody. The polyclonal antibody means that antibody having that antibody, the one protein blocked, it interact with the n number of the uh, n number of antibodies, that are polyclonal antibodies. The small peptide immunogen yield fewer restricted epitope polyclonal. Here it's only one. This particular portion is this interaction portion is removed, and it indicated by the that also bound with the another antibody. Then isolate and fuse the B cells to the hybridoma cell line. Here we obtained one new kind of the antibody interaction. We want to insert the into one hybridoma cell lines. That hybridoma cell line produce the monoclonal antibody. This is the laboratory scale experiment. First we want to inject the antigen, that once entering the foreign particle in animal, that immune system is activated, that is a B cell activation. So that B cells are isolated from the animal. Antibody forming cells, that are the B cells, that cells are fused with the hybridoma cell. Hybridoma cell is the empty cell without nucleus, that can be grown in multiple count. That is empty cell, that hybridoma cells fused with the hybridoma cells. It's a very important step in monoclonal antibody production. First inject the antigen into the animal, that antigen immune system get alert, that producing that B cells are isolated, and B cells are fused with the nucleus free cell. So this hybridoma cell is formed. After the hybridoma cell formation, that is producing antibody, and it cloned that cells also divided in a larger number. So there is a n number of antibody production happen in the medium. Then we can isolate the monoclonal antibodies. These are all the steps involved in the hybridoma technology. It's one another technique happen in the rabbit. Once inject the antigen in the rabbit, then antigen B cells are recognized the antigen, and B cells can develop the ability to facing the antigen, so that are producing the memory cells. Memory cells means okay, this antigen having the this active sites. Once it's enter into the body, we can block this antigen with the antibody. This is the basic concept. The memory cells means it's for the future purpose. Once the B cell get affected by the antigen, that B cell are producing the receptor for the blocking inhibitor for antigen. So in this step, the B cell recognize okay, this antigen having the triangle shape blocking side. Now if we design the weapon to the antibody to block, this means we can control the infection, so that memory cells are get alert and store the memory for the future purpose. So only here you can see this B cells having the triangle shape receptor receivers. Okay, after the experiment we can collect all kind of the monoclonal antibodies, secretion of the antibodies, then inject it again into the that human cell. This is the kind of it's a one basic experiment which explain the plasma therapy. The plasma cells having the B cells, that B cells are having the antibodies against the viral infection. This so the plasmas are injected into the another patient. It will helps to cure the develop the immune system in the patients. Next immunological, first immunology technique we discussed is the antibody, monoclonal antibody production. Why we want to produce the monoclonal antibody? We can apply for the medical purpose, medical therapeutics and the diagnosis purpose. The first time I explained the rabbit experiment, it's a therapeutic application. Once we want to develop a memory, that memory helps to block the future infection, that is the therapeutic, or it may be help us to treat another patients. In the experiment we are going to explain the monoclonal antibodies with the deduction diagnosis methods. Okay, in this ELISA, first it's an sandwich ELISA experimental method. No, it's a direct interaction. In this image they explain how the antibody get interact with the antigen. First step is a antibody placed in the medium. Okay, how they produce this antibody? Same as explained in the previous slide. They are produced the monoclonal. They produced the artificially. They are producing the mono antibody. That antibody is coated on the surface. Once we are adding the test sample. The test sample is the antigen. It bound with the if that having the specific site for the interaction that bound at the that antibody. Then after the section we want to after the bound antigen antibody interaction, we want to understand the that bonding with the enzyme linked deduction antibody. That is the another antibody, secondary antibody is added on the antigen that having the enzyme link. This enzyme having the once after the bounding of the enzyme linked antibody on antigen, that is in inactivated form. Once adding the substrate to the enzyme, that will get that expressed in OD value. Otherwise, without this if there is no antigen interaction, there is no enzyme linked antibody binding, so there is the no enzyme activation, so there is no color. It's an experimental output comparison. Once that experiment, your sample having antigen that producing that activating the enzyme substrate activity, that enzyme substrate enzyme substrate activity helps to produce the color or intensity, that OD value. Otherwise it not produce the, there is no bonding happen in the top portion, so there is no enzyme conversion, so there is no OD or color change value. Here we have a different type. One is the direct method. Antigens are present in a sample, we want to add the enzyme linked antibody. This antibody gives a color. Once after the binding, we want to do the wash. Other antigens are removed from the sample. Once if there is a interaction with a monoclonal that antibody and antigen, there was a enzyme. That enzyme is activated with the substrate that gives a color. Each and every step we want, washing is the very important thing in the direct in the sandwich ELISA. Here First, the director is just add antigen sample addition, then add the antibody, then add the substrate. It's all, no need to do the washing. But sandwich ELISA, the washing is important step. First, primarily that sample kit is coated with the antibody. Then we want to add the sample. Here, please understand. First step, we want to add the coated the kit, that store that plate with the antibody. Then only we want to add the sample. If there is any recognizable antigen in the sample that bound with the placed antibody, that is orange color. After this, and after that binding, we want to add the primary antibody. That addition of the another secondary antibody. This secondary antibody help us to understand are recognizing the antigen only specific for the antigen. If there is an antigen, it bound at the particular portion. After that, we want to do the wash. If there is interaction happen, means that antigen antibody and the enzyme linked antibody was there, that produce the gives the expression for the substrate. So only that final yellow color happen. Then competitive, another one is a competitive. First one is addition of the antibody. Then the second step, we are adding the antigen and antibody, are both in the same step. If there is antibody interaction happen in the sample, okay, that sample having the immune effect, this is happen in our rapid test. Okay, that they are they are coated the kit with this that antibody. That's our human sample having the IgM or IgG, means that is interact with the base antibody that producing the enzyme activity, that color change. First one is once again repeat. First one is the sample addition, then only add the monoclonal antibody, then only add the detection antibody. Then sandwich ELISA. In this sandwich ELISA, first step is we are coated with the antibody, then only add the antigen. Another step is a competitive inhibitor. In this competitive inhibitor, we are having the antibody on the coated plate, then add the sample, that sample having the immune response against the antigen that is recognized by the placed antibody, this producing the expression. These are all the ELISA method which is going to detect the disease, that is the detection diagnosis the purpose. Another one, so here I am once I am explain with the competitive, because it's an image which helps to understand. Okay, first is a sample addition to the monoclonal. Here that's they have mentioned the this red color is a placed antigen. Okay, then add the antibodies with the enzyme, that if there is an interaction happens, that enzyme activity, that may be the antigen and antibody. Okay, next is a radioimmunoassay. Radioimmunoassay, it's a radioactive linked, right? Here we are going to explain about the fluorescence molecule labeled. The enzyme labeling and fluorescence molecule labeling is a very very important thing in the addition of the radioactive molecules in the experiment is very careful. We want to carefully handling the radioactive molecules. Here they understand the interaction, understand their explain the output with the radioactive molecule. First, for that we need antigen, antibody and radioactive label against the antigen. Against the antigen means it's a favor to antibody. Here the one important thing, in the radioimmunoassay we need to do the antigen, antibody. We need antigen, antibody and a radioactive label. This radioactive label want to against against to the antigen. That means it's a favor of antibody. Okay, it's having a favor structure, right? Here they added the in mixture and give some time to reaction. After the reaction, we are remove the excess molecule, separate the molecule, then collect the bounded molecule based on their that density variation. Once it's very it's a small, it's a antibody alone means it's shown some molecular weight. It's bound with the radioactive molecule, and antigen means it gives some molecular weight. It's vary between the native and the bound compound. Here, after the bound fraction collection, we are explain the we are understand the immunoassay with color change. Here it's a principle. Here it's using the antigen antibody binding. It's based on the antigen antibody binding, we can understand the quantifying the amount of antigen antibody interaction. Based on the this antigen antibody interaction is identified using the radioactivity. It's a procedure. First radioactive agent, first antibody. It's a first primary antibody, then antigen added to the sample. Here it happened, right? Antigen antibody interaction happen in the cells. That is the also the antibody is primary antibody is loaded with the radioactive antigen. During this reaction, this radioactive reagent replaced with the antigen. So this radioactive compounds are freely available on the sample. Then that antigens are bound at the antibody. Here it's an explanation. Then by adding the secondary antibody, that primary antibodies are loaded on the secondary antibody. This after this secondary antibody addition gives some reaction, the time for reaction. After that, based on the color intensity, we can explain the how much amount of antigen present on the present in the sample. They have a standard value because they know how much amount of primary antibody they added. For this much of primary antibody, they want to produce this much of color. But after the addition of antigen, there was replacement happen in the sample. This replacement reduced the intensity color of the radioactive antigen. So in this after the addition, secondary antibody addition, there was some small amount of changes, that replacement happened, right, in the primary antibody. It's expressed as a color intensity variation. Okay, next this radioactive, apart from the, that is one kind of the radioactivity method. Another one is the Western blotting. In this Western blotting, we have the sequential step. First, we want to do the electrophoresis with the gel. Then we want to do the membrane transfer. In this membrane transfer, in between the gel want to placed in the between region, that membranes are placed over the in top and bottom of the gel is that bottom top of the gel is placed with the membrane. Then we want to place the sponge. Why we want to do this means we want to transfer the DNA, that transfer the proteins in the that proteins means antigen antibody molecules. Okay, transfer the proteins, that is the antigen. Transfer the protein from the gel to the membrane. So this setup is help us to do the transfer process. Then blocking means that we want to avoid the movement of protein in the membrane. So we are doing the blocking with primary antibody incubation. After the primary antibody incubation, we are if there is a specific interaction happen in the membrane, that antibodies are placed over the membrane. Then secondary antibody is added specific for the primary antibody. If there is interaction happen, means that gives the band in the image, that membrane color change, that is the membrane. We can we can understand the binding, understand the protein antigen interaction, antigen antibody interaction through the western blotting membrane. So here in the western blotting method, the membrane is a very important thing, which helps with the each and every step. We want to carefully handle the membrane, because this membrane carries the protein. Once again, I am repeat with another image. It's an gel, that is proteins are separated, that antigens are separated. Then we want to do the plotting. We want to do the protein transfer to nitrocellulose membrane. This nitrocellulose membrane having the uh, once first place the gel, and then place membrane over the gel, then place the sponge. It's a setup. Here they mention, right, they make some impression, that impression, and they are adding in this blotting, this protein transfer, that the DNA is directly transferred to the membrane, nitrocellulose membrane. So after the transfer, that membrane is transferred into this immunoblotting instrument. This is the western blotting instrument. It looks like a bottle. In this bottle, we want to place the that membrane. It's a rolling bottle. In the experiment, it's rapidly scrolled in the instrument. After in this step, we have the antigen. In this step itself, we are added the first step. They are added the primary antibody. After that, doing the washing. Then secondary antibody addition. Then deduction. It's a final step for the autoradiography deduction. This develops the this autoradiography is understood by the band appear on the membrane. If there is interaction happen, means it gives some color. Sometimes it may be the blue color, it may be the red color. It depends on the antigen antibody interaction, that secondary antibody activity. Then immunoprecipitation. In this immunoprecipitation, we can understand the immunoprecipitation with the antigen antibody. Once the sample having the antigen, that the antigen is interacted with the antibody. That antibody then by addition of the substrate on the or a binding molecule for the antibody, that due to the molecular weight, higher molecular weight, it settle down on the settled out in the cell, in the solution. After that certification, we can obtain the precipitator molecule. It's another strategy in the immunoprecipitation. The recombinant cells having the different antibodies on the cell surface, that we are collected the antibody, then incubated with the that antibody antigen. If there is specific antibody for the antigen, means that gives the interaction. Then after the interaction, we want to do the STS phase. This is the STS phase. Cells bonding, it's explained whether it is having the antigen antibody interaction. Okay, next one thing we discussed, the diagnosis therapeutic method, diagnosis method, then immunoprecipitation and immunoassays related ELISA techniques. Now we are entering into the advanced techniques in the immunology, that is the flow cytometry. This flow cytometry is the very important one in the today's experiment. today's all the kind of researchers are doing this flow cytometry for their better understanding. Okay, what they did. Okay, what is the color? First, we want to understand what is the color in the in this image. Okay, first we have a mixture of cell. We are adding the antibody. If there is a non-target cell, that is a it's unwanted cell, means that explained as the black color. If there is a target cell, target cell means that cell want to have the the cell want to have the antigen on the surface. So is that the target cell interaction happen to antibody, it gives the color change, that is the red color. Okay, here in this image they added the sample. Okay, in this sample, if there is the optical laser recognizing the that molecule, means it gives a signal to the detector, so that we can understand how much amount of recombinant, that specific cell present over there. In this step we can collect the specific cells also, that is sorting. In this flow cytometry instruments, first they added the group of sample. In the path it allows only one cell alone. Okay, if there is a needed cell, if they fixed the wavelength. Okay, for example I mentioned, if there is an red signal for target cell, means okay, detector recognizing the signal, that's color intensity, that detector activating the sorting machine means collecting that. Here in this bucket it's shown the waste material. If there is if there is a need of target cell, means it moves and place the that collecting tube, collecting a specific cell, that is a cell sorting. Here they mentioned as it's how it detect. Here is the laser beam formation. Here they pass the sample. This sample is having various receptor on cell surface. It's expressing as a various signals to the detectors. These detectors are transfer amplifying the signal into the digital conversion. This digital conversion given as a input to the system. Here how the cells are cells are understood and recognized on the cell surface. Okay, in this step they having the group of cells. That group of cells are having antigen fluorescent dye on the cell surface, right? This fluorescent dye on the cell surface, this fluorescent dye is the recognition molecule. If there is a fluorescence happen, means okay, that cell is we want that cell, otherwise you don't want the cell. Okay, in this step detector recognizing the that fluorescence dye, then that's move down. In that step we have a positive and negative charge on the cell surface. If there is a fluorescent dye, means it carries the power charge. Based on the charge interaction we can separate the cells. Here we can understand cell surface having the negative charge. Here they may placed the electromagnetic rods. These electromagnetic rods are recognized the anti gen and that recognized the targeted cell. The targeted cells are collected in the separate tube. It's an experimental methods. First addition of the target cell, it's recognized by the detector, then it's moved towards the end of the pipe. At the end of the pipe it separated based on their charge on the surface. It's a fluorescence immunofluorescence microscopy. In this immunofluorescence, it's a very important and simple concept. In this, we have the light source, excitation filter, dichroic mirror, this then emission filter. Okay, these are the four important parts which help us to develop the image for immunofluorescence microscopy. In this image, it's not it's a basic image, because that here the specimen placed over the stage. Here one in this image, that objective is observed, it's it's producing the signal, it's observing the light source, right? Here they are producing the light source. This light source is this deviated by the dichroic mirror. This mirror helps to pass the light on objective. This objective, okay, this object is receiving the light, means it's producing producing the fluorescence. Otherwise it's not producing the fluorescence. If there is a fluorescence emitted on the cell surface, means that cell surface having the antibody. It's an understanding based on the experiment. Here we have that's a plot, the cell surface having the antibody with that fluorescence molecule. Once you pass the light, it emit the fluorescence. It's another better explanation. Here you pass the white light on the specimen. The specimen surface having the antibody with the fluorescence molecule, so it's pass the fluorescence to your visibility eye. And then another important technique is the FIS and GIS. FIS means the fluorescence in tissue hybridization. Then another one is the genomic in tissue hybridization. Here in tissue hybridization means the modification, it's a modification modified technique. In situ hybridization means inside of the in situ hybridization is located of the gene or DNA visualized by the chromosome, the chromophores, that all fluorescence molecule. Due to the molecule we can detect fluorescent molecule deposit happen in the fluorescence in tissue hybridization. The another thing, genome hybridization used in the genome in tissue hybridization. This is the one best example. First explaining image, first step they are having the DNA probe is labeled with the molecule. This indirectly added to the slide. Then here we having the non-labeled DNA. Here we have the labeled DNA, here we have the non-labeled DNA. Then did the denaturation happen in the both labeled and non-labeled DNA. In this denaturation in tissue hybridization happens. What is in tissue hybridization happens? The non-labeled DNA is recombinated with the labeled DNA. Here it's happened. It's a basic understandable. Okay, after this recombination, the produced DNA can detect the fluorescence. That produced DNA is having the ability of the interaction. It's a one important step. Here another one is the unlabeled normal hybridization or normal hybridization. Here we produced the chromosome with the DNA with the labeled molecule. Here we have the chromosome with the non-labeled molecule. It's visualized under the slide under the microscope. If there is it help us to detect the sample, how much amount of hybridization happened. It's a schematic representation. Here there was the gene. That gene is randomly cut down by the DNase enzyme. After that, we added the DNTPs. This DNTPs are denatured. This double strand is denatured at 72 degrees Celsius. Here another side we have the fixed cell. This cell having the DNA. This normal DNA. This normal DNA is denatured. This denatured normal DNA and denatured modified DNA is hybridized here. This hybridization, this modified DNA that have DNTPs having the biotin label. This biotin label help us to help us to recognize the antibody, specific antibody, that is the avidin, biotin, avidin. They both are the help us to explain the fluorescence into hybridization. Here that these CNTPs are labeled with biotin. This biotin labels are recognizing the avidin linked fluorophores. Here this yellow color is a fluorophore. This avidin. This biotin avidin interaction makes the fluorescence. If there is a biotin avidin interaction happen, means that fluorescence get activated. It produces the fluorescence. Otherwise, it not producing the fluorescence. The main focus is understanding of the recombinant and understanding of the modified understanding of the produced fixed cell. Here it's an detector experiment. Here this is called as the basic fixed cell experiment. Here it's a normal cell having the DNA, that chromosome that's denatured at 42 degrees Celsius. Another thing, another side we have a gene. That gene is modified with the biotin label. This biotin labeled DNTPs are undergone denaturation. This after this upper strand is overlapped with this down strand, that's lower strand. These two strands are forming the double strand, that 3' strand of the modified gene and 5' strand of the normal cell is incorporated and producing the fluorescence, producing the signal for attraction of the avidin. This antibody and antigen interaction producing the fluorescence, that producing the fluorescence under the microscope. Here this is the genome hybridization. First image itself I mentioned the one probe DNA, it interact directly labeled and indirectly labeled. Here this is directly labeled. Directly fluorescent molecules are attached. Here we are added the antibodies for the fluorescence. The denaturation happens. This denatured two strands are incorporated with the block, that's normal DNA. This normal DNA chromosome is formed, and this formed DNAs are having the directly labeled molecule and indirectly labeled molecule. In this structure we can see the probe with the signal, denatured signal, that directly labeled signal and the indirectly labeled signal on the surface. This is the incorporation. This is the hybridization, that's making the hybrid of chromosome level. Then we are entered into the biophysics concept. This biophysics concept, we having the NMR microscopy, IR spectroscopy, UV visible spectroscopy. Why we want to this different value? Based on the spectrum, electromagnetic spectrum range, we want to vary the spectroscopic method. If there is the molecule is visible, it can be absorbed by UV light, means we want to use the UV visible. Otherwise it is that having the specific IR radiation, IR wavelength, means we want to understand the image, that structure with the IR spectroscopy. that is the FTIR spectroscopy method. Another one is the NMR spectroscopy. It's a radio waves. Based on the radio waves, we can understand the NMR spectroscopy. What is the information obtained from the NMR? It's a specific arrangement of all carbon and hydrogen atoms in the compound. We can understand the arrangement of carbon and hydrogen atoms on the compound. Another one is the functional group present. Here IR spectroscopy. This FTIR spectroscopy is fully depends on the functional group present on the cell surface. Please understand. This is a very important topic that they rapidly ask the question in the CSIR exams. Only I mentioned this. The NMR spectroscopy is activated. Principle is based on the radio waves. This helps to understand the arrangement of all carbon and hydrogen atoms. In the IR spectroscopy, that is FTIR spectroscopy, IR spectroscopies help us to find out the functional groups of the compound. In the UV visible spectroscopy, we can identify any conjugated pi systems present in the compound. Is there any interaction happen in the compound? The compound normal basic compound interactions can identified by the UV visible spectroscopy. Okay. It's a principle of the UV spectroscopy. Okay, we have 30 more slides. Shall we go ahead or continue with tomorrow? Or shall we continue in tomorrow class? Because I want to clarify with you. Please tell whether we want to confirm or shall we go that with tomorrow class. I have 30 more slides because it's a vast topic. We have two topics, biophysics and radio labeling. Participants, please let me know. Shall we continue or yeah, thank you so much. Thank you for your response. Okay, it's a UV visible spectrometer. In this, T2 lamp help us to detect the molecules, some molecules under the wavelength 400 nanometer, and that kind of we can change the light for visible and UV. This light source is maybe the D2. D2 means you observe the UV compounds. It's below 400. Below 400, it is UV wave. Below 400 nanometer is the UV wavelength. This UV wavelength recognized by the D2 lamp, deuterium lamp. It's a very important thing. There is a basic thing. They are asked the question with a critical concept. But the one thing is UV visible, UV nanometer below 400. 200 to 400 is the UV wavelength. This 200 nanometer to 400 nanometer range is absorbed and recognized by the deuterium lamp. Another one is the above 400 to 700, the 900, 700. That's 700 is the major maximum fix. 400 to 700 nanometer is absorbed by the tungsten lamp. This tungsten lamp is helps to recognize the visible range molecule. It's a two important thing we want to understand in the spectrophotometer. Okay, they are provided this question like this compound having the 400 nanometer absorbance. 400 nanometer absorbance means what is the light it helps to recognize the that compound in the spectrophotometer. For that kind of question, we want to understand this basic concept. If that molecule having the 400 nanometer recognized absorption means it can be it can be captured by the tungsten lamp. If they are provided that compound having the 250 nanometer absorbance means that able to detect under the deuterium lamp. 200 to 400 is the UV range. For UV range we want to use the tungsten deuterium D2 lamp. 400 to 700 is a visible range. For that visible range compounds we want to use the tungsten lamp. 200 to 400 we want to use the deuterium lamp D2. 200 to 400 it's a visible UV range. 200 to 400 is a visible range, we want to use the deuterium lamp. 400 to 700 is a visible range, we want to use the tungsten lamp. Another thing is the after that absorbance, that molecule here it's this mirror, it's reflect the reflect the light, the diffraction grading. After that light pass through the filter to mirror 2. This mirror 2 reflect the image to mirror 4. This mirror 4 and transfer the image to the lights to the detector 2. Okay, next is the it's another basic example of the fluorescence spectroscopy, fluorescence microscopical image. In this fluorescence microscopical image, here there is a sample state. We place the sample over there. It's a light is passed through the this particular portion. This after the light absorbance on the cells objective, that objective producing the fluorescence light, that fluorescence light absorbed by the spectrophotometer, and this spectrophotometer it's emit the transfer the signals to the monitor. It's a circular dichromism spectroscopy. It's a very important thing. First is the spectrophotometer. The spectrophotometer helps to detect the visible and UV absorbance molecule. Then is a fluorescence spectroscopy. Fluorescence spectroscopy, it's another technique which helps to develop the develop the images with the fluorescence absorbance. Another thing is a circular dichroism CD spectroscopy. This CD spectroscopy is an absorption spectroscopy. CD spectroscopy is an absorption spectroscopy. It having the different parts. It's a light source. This light source pass the through the filter wheels. This filter wheels filter the light and pass to the that cuvette having the sample, that is helps to understand the protein structure. All the things are happen with the wavelength understanding. Here wavelength, psi wavelength, you all studied. These are all the sine waves. Using this sine waves we can do the experiments. Okay, this is a principle of the circular dichroism. Okay. Yeah, okay, fine. I'm just finishing this spectroscopy topic alone, that biophysics topic alone. Tomorrow I will continue. Here why I am mentioning this means, based on the absorption we are get the wavelength for peak for the wavelength. The peak wavelengths are converted to dichroism. Here that is the concept. It's a very simple technique which understand the molecules, the photon. Using the photon beams we can understand the molecules. Here they mentioned the complicated protein. So that based on the protein observance, we get the that modified sequence. It's very simple. So based on the image, based on the structure, we can understand the compound. Here the left polarized light is passed to and right polarized light. Here the between the signals observed by the two lights, here they mentioned here different lights are passed, right? Based on the light intensity we are observed the signals. Difference between the signals are helps to understand the structure. Then another one is the NMR concept. This NMR concept is a very important thing. It's also absorption of electromagnetic radiations between from the 4 megahertz to 9900 megahertz. It's quoted one. NMR microscopy is a spectroscopy is a absorption electromagnetic radiation based techniques. In this technique we having the it can it absorb the radiation between the 4 megahertz to 900 megahertz. This radiation absorbed by the nucleus or nuclei of the compound. It may be the cell, it may be the compound. Every compound having the nuclei, right? That nuclei at the nuclear level we can understand. So only they mention that helps to understand the atoms of the carbons and hydrogens. Two types of waves are used in the NMR, that is a continuous waves and pulse waves. It's a setup. Sampling tube is there. That the magnetic electromagnetic waves are passed to the sample. That electromagnetic waves helps to detect by the detector, that producing the signals to printers. That based on the electromagnetic signal on the atom, we will develop the signal. It's another NMR technique to understand the protein structure. Here that NMR protein sample is loaded in the tube. This sample is transferred into the spectrophotometer, that is the NMR spectrophotometer. That setup produce the signals like this kind of this kind of peaks. That these peaks are undergone the data acquisition. Data acquisition means it's converted into the signals. These signals are helps to produce the image protein structure. Here these peaks are the atom peaks. Okay, C having the height, this much height. Position of the atoms. This peak helps to understand position of the carbon atom and hydrogen atom, and it helps to produce the data acquisition. This after the acquisition obtaining, we develop the image for the protein structure. Then electron spinning resonance spectroscopy. The electron spinning resonance spectroscopy, it's very important thing, is based on the electron transfer. We doing this experiment. It helps to understand the transient metal complex. Previously we discussed the protein. It helps to NMR helps to the compound and the protein. Spectroscopy also helps to understand the this dichromosome helps to understand the protein structure. Then this spectroscopy helps to understand the compounds. We can measure the protein also, but we can't find the structure. Fluorescence, it's depends on the antigen antibody interaction, and the fluorescence molecule present on the structure we can develop. Dichromosome helps to understand the proteins molecules, and then NMR helps to understand at the nuclear level. It may be the protein, it may be the compound. Here the spectroscopic method helps to ion metal complex. It helps to understand the geometrics in the crystals. Here there is a absorption spectrum of the compound. It's the electron spinning microscopic spectrum is look like this. Here we can measure the different ion complex. We can find the inorganic compounds. All the things will happen in the uh, electron spinning microscopy. In the biological application, they are used for the detection of the sulfur, copper, free radicals. These are the four different optical spectroscopy comparison. If there is absorption microscopy, its absorb or ratio of transmitted incident radiant power. It's an measured quantity. Its absorbance or the ratio of the transmittant. Absorbance or the ratio of the transmittant. It's the output. Its example is the UV IR radiation spectroscopy, atomic absorption microscopy. Emission based technique means the radio power of the emission. This radio power of the emission is understand the that ICP, the induction coupled plasma microscopy. Another thing is the dichromism or the emissions based experiment. Luminescence experiment, ratio power of the luminescence. This is molecular fluorescence molecule using the molecular fluorescence and the fluorophores. We can understand scattering. Using the Raman scattering, we can do the Raman spectroscopic experiment. These are the different X-ray crystallographic method, electron microscopic method. This X-ray molecules are recognized by the X-ray crystallographic techniques, electron microscopy, then electron probe microanalysis. The ions are the secondary ions in the mass spectroscopy analysis, helium ion spectroscopy. These are all the spectrometric method. This is X-ray crystallography technique. In this X-ray crystallography, first we want to develop the crystals, then observe under the X-ray diffraction X-ray diffraction microscopy. That based on the electron density, they develop the structure. It's the basic techniques. The rotation of the anode, this having the electron beam. It's this having the structure. This anode having the structure. The copper is the anode, rotating anode. The primary beam is passed to the focusing mirror that observe on the crystal. Here we placed the crystal. This is our sample. It's an source, the copper source producing the signal based on the electron magnetic resonance. The X-ray based on the electron absorbance that produces signals to the mirror. That signals are absorbed by the crystal structure. This crystal based on the intensity, the electron density. If that is having the higher density, that producing small the higher dot. The based on the density. It's a very very important term. And based on the electron density in the crystal, we can find the structure. First we produce the dot image itself. That dot images are helps to develop the image, develop the protein structure. Here again mismatched one. It's a first thing of I'm in this step I mentioned. Sorry, I missed. These are the techniques helps to understand the protein structure. X-ray crystallography and NMR crystallography. So only I am secondly repeating this NMR. Sample preparation, sequential assignment, side chain assignment, and then collection of the NMR restraints, NMR sample structure collection, and structure validation. This first you want to understand the atom level, then amino acid level, then structure. That second is structure, loops, region, hydrophilic region, hydrophobic region, that kind of understanding, then only final structure is developed. It's an scattering light microscopy, is Raman microscopy. Okay, this Raman microscopy is this scattering light microscopy is basic thing, having the Raman scatter light, Rayleigh scatter light, and then Raman scatter light with the emission values. Why I have mentioned this image means scattering microscopy is very scattering light microscopy is are working with this light scattering of the molecule, and this effect helps to Raman Raman spectroscopy. This Raman spectroscopy, they are passing the laser light to the molecule. These molecules are vibrated. Based on the vibration, that producing the Rayleigh scattering, that scattered images are observed by the diffraction grading. There is no light. Based on the vibration alone, the detector is recognizing the signal. Okay, this is the another image. Here the light is passed to the molecule. That molecule give the some shaking on the particular screen. That shaking is recognized by the this photodiode. So only there is a molecule. That molecules are having the shaking, that moving, that I explained with the larger particles. Then DLS, that this DLS is the particle size analyzer. This dissolved this helps to understand the particle size, particle movement. It's everything happen because in the DLS instrument we having the cuvette like instrument. In that cuvette we place the sample with the solution. In the solution, that Brownian motion, based on the Brownian motion of the molecule, it developed the image, that developed the okay, this density, this much amount of particles are there here. It's a detector, this a cuvette which we pass the laser to this cuvette. In this cuvette there's the particles, right? That particles are passing the signals, and it helps to produce the theta potential. It's based on the theta potential value. We can tell the particle size. Using the DLS, we can understand the theta potential and particle size. It's an ionization process. In this ionization process, it absorbs the light. It's ionic deuterium lamp is on passed through the sample. That samples are based on the that both side interaction, that are aligned in the line. That aligned molecules are separated by the this particular screen. That separated molecules are linearly passed through the detector, the electron multiplier. That multiplier helps to develop the and it multipliers are helps to form the reflected mode of the molecules, that absorbed at the reflectron mode. This is a portion of the DLS. Here that is the detection part. In this detection part we having the we are having the primary ions and secondary ions. In this primary ion secondary ion absorption, that energy is vary, because primary ions are passed on the sample, that produce some energy ions. That the ions are quantitatively analyzed at the energy analyzer. This energy analyzer, it's pass the signal to mass spectrophotometer. This mass spectrophotometer is attached to the detector. This based on the signals. Here we having three type of signals. That is the mass spectrometric peak. This peak value compared with the standard and detect the mass of the particular compound. It's a setup of the compound. Here we inject the sample. In this portion we can inject the sample. The electron source is there. This electron beam is passed on the sample. The particles are accelerated into the magnetic field. Here this is the magnetic field. After this magnetic field entry, it's charged with the particle beams. Okay, then magnetic field is separated. Using this magnetic field, this image itself they mention, after reaching this this magnetic field, this molecule gets separated. The separated molecules are move based on the weight, higher molecular weight and lower molecular weight. If that is a light molecular weight, it's past it's absorbed by one area that enter into the another particular portion of the detector. If there is a higher molecular weight, it's move slowly and reach the detector at the particular temperature. It's a MALDI-TOF analysis. MALDI-TOF analysis help us to detect the protein structure. MALDI-TOF. Every protein structures can be detected by the MALDI-TOF. Here we having the desorption. This desorption means the laser beams are attached on the protein. Here there is a protein. After the dissolution of ionization process, that molecules are detached and that particular protein alone detected. The MALDI-TOF technique is a very important technique. The protein, purified protein, it can be detected by the laser beam. That it's detected based on the MALDI-TOF structure, and your collected samples are detected and shaped, and structure of the compound is detected based on the MALDI-TOF analysis. Here the photon transfer happens in the during the ionization process. In this ionization process we can explain the total mass analysis. Then next is the LCMS. The same principle of the GCMS happen in the LCMS process, because another thing is here they use the liquid. That different thing is the LCMS process is that they use the liquid as a molecule. Then another is the surface sensitive surface plasmon resonance spectroscopy. In this method helps to detect the surface sensitivity of the protein. So we can understand the protein interaction, we can develop protein ligand interaction, we can understand the protein DNA interaction, protein membrane interaction. We can the protein related of all studies, the surface related all studies, because cell surface having the n number of proteins. This surface protein related topics are covered, that are identified by the surface plasma resonance spectroscopy. The light energy is interact with the delocalized electron, that is the surface electron in the metal surface, that reduce the reflector light intensity. Once the light is absorbed on the cell surface, means that the intensity will automatically reduce. It help us to understand the that light intensity variation. It help us to understand the surface modification. Here the light source are passed on the cell surface. The samples flow on the cell surface. If there is the interaction happens, means that absorb the light, that producing the less signal. There is no interaction happen in this particular portion, so there is less intensity, less light alone absorbed on the surface, so light intensity output is high. Here that blue color, that yellow color means that is the less intensity, that that yellow color means that is the light passing. The light, imagine this yellow color means that light rays. Okay, here you pass the light, there is no interaction, no molecule is there, so that light directly emitted, that is directly absorbed by the detector unit. If there is an interaction, means there is light is absorbed by the interaction, that is the high density molecule, we can't receiving the light. is a sensor based technique. Using this surface resonance surface plasma resonance, we can using the sensor develop the sensors. It's a gold chip. This gold chip is coated with the peptides. These peptides are specific for the antibodies. This antibody, the immunological antibody, this technique is alone used for this laser kit, coronavirus detection kit. So only I am included this image for the surface resonance spectroscopic method. This basic method they are using the kit. Here they are using the spectroscopic method. They are detected based on the image, that color change alone. This in this step itself the color change happen after the antibody binding, antigen antibody binding. There was a color change happen on the golden base, that kit. After that we can quantifying using the detector, the surface plasma resonance molecule. Okay, then this radioactive labeling technique. First we want to understand the properties of radioisotopes. Why we want to use the radioisotopes? This radioisotopes are helps to detect the it can kill the cell, it can mutate the cell, it can be having different properties. Because that radioactive labels are very sensitive, we want to that it may be cause the cancer. So if you are working with the radioactive isotopes means we want to be careful. The same chemical properties as non-radioactive isotopes, the same elements, every properties are same, but the sensitive, that toxicity is higher than the normal isotopes. Okay, decrease with the time. The radioactive detection time is very less. Here they mentioned three types of the particles: alpha particles, beta particles, the gamma particles. This each particles is 2 protons and 2 neutrons elements, that is called as the alpha particle. These particles are the electrons. Beta particles are the electrons. That gamma rays are the electromagnetic waves, that similar to the X-rays. First one is that alpha particles are similar to the identical to a nucleus of the helium-4. Why I am mentioned this means based on this we can develop the experiment. Okay, if you want to do the experiment related detection method using the helium means you can develop the alpha particle. You can take the alpha particle. If you want to relate the X-ray related molecule means we want to detect the X-ray related experiment. We want to do the X-ray alternative to the X-ray molecule means X-ray experiment. We want to select the gamma rays experiment, gamma rays particle. Why, as only mentioned, this alpha particles are not able to penetrating. This beta is moderately penetrating to the things, but gamma rays are very penetrating. So only they are using this replacement of the X-ray. X-ray is penetrated to the bones, so only they are using the X-ray. So likewise it's having the highly penetrating property. Here I am going to explain the just 2 minutes I am going to finish up. Sorry for the I am taking too much of time today. Sunday I am not expecting this much time taken for this session. Sorry for the sorry for this delay. Just 2 minutes I am going to end up. This carbon 14, it helps us to the research process. In this research purpose we can develop the determine the steps involved in the photosynthesis. If we are doing the research on the plant photosynthesis, we can use this carbon 14 radioisotope. This phosphorus 32 and 33 is used to the genetic level. Cesium-75 is the protein studies in the life science. The strontium helps to metabolism and bone formation. Hydrogen 3 and tritium is used to the life science studies of the drug metabolisms. Then Cobalt 16 radiotherapy, the radiotherapy and prevent cancer. This is the one important thing. Every hospitals having these cobalt isotopes, they are following the safety levels. Then iodine the 131, the local brain tumor, we can treat this with the local brain tumor. It's a very high expensive one. For one nanosecond some time period, they are asking the 5 lakh or 7 lakh some amount. The carbon 14 in the medicine field, they use anemia patients are used. Carbon 11 is a used in the PET scan. Sodium 24 is a blood circulation studies. Thallium is 201 is helps to use the heart tissues damage, determine the heart tissue damage. Yeah, it's a auto oh no angio test. In the heart attack patient, they did the angio test that the detector having the this thallium 201 compound. Then in the industrial level, they use the americium 241. It helps to find the uniform thickness when rolling the paper or steel. Determine the location of the oil well. It's a very important thing that determine the location of the oil well is we can determine using the americium 241. The sodium 24 is help us to the detect the location leakage of the pipelines. Leakage of the pipelines are detected using the sodium 24. Then deuterium is helps to aircraft part, develop the aircraft, the boilers and integrity of the boilers and aircraft parts. Uranium, it's a nuclear power plant, you all know. Californium is detect the moisture content of the soil. The americium is a very important, and the sodium 24 is a very important thing. These are all I mentioned. Repeatedly I mentioned that compound. Here I mentioned the half-life. The half-life based question is a very important. The carbon is a high half-life. Carbon 14 having the higher half-life, that is the 5730 years. 5730 years. And the technetium is having the 6 hours. That having the many applications. Here a molecular imaging process, they are in each and the isotopes also used for the molecular imaging. That is the PET instrument. That PET instrument means the positron emission tomography. The positron emission tomography, it's an MRI, the MRI scan, that kind of scanning scanning CT scan is combined with the positron emission tomography. This using the rays previously mentioned, right? Alpha rays, alpha, beta, gamma rays. Based on the penetration, they are differentiate. That beta and the gamma is penetratable. That gamma is very penetratable. Here they are using the gamma radiations, the isotopes. It's help us to understanding the image of the organs. Okay, diagnosis of cancer. If there is a one part having the cancer in the human body, just pass the rays means we can understand the size of the cancer. We can understand the block happen in the cardiac. Okay, there was a big hole, big accumulation happen in the cardiac. That kind of things. Images are recognized by the PET, the positron emission tomography. It's a basic principle how it's activated. Thank you so much, and sorry. Today's section I am going to finish up with biophysics techniques, radioisotope technique, and the immunological techniques. We have finished the almost the main techniques. We are finished in the molecular methods in biology. Tomorrow we will discuss the statistical method, microscopical technique, and electrophysiological method field in the biology. Thank you so much, everyone. If you have any questions, please post it, and I am not tell it in this session. I will post usually I will post in the your Google Classroom and mail IDs. If you have any questions, please post in a chat box. I will. If you have any questions in the session, just post it in the command line. I will explain the answer in the Google Classroom and mail IDs. Thank you, everyone.